exam 2. And so you should be able to, when you go and look at your attempt, you should be able to see the right see the right answers for the multiple choice, and also for the essays, right? Like I always make sure I write in the correct answers and the general feedback. So you guys should be able to see that. So just let me know if you can't. You also should be able to see your grade in the course, you know, as it stands right now, and it's up to date and accurate, right? I mean, Moodle is coded exactly as the grading policy is listed. So if you see your grade on Moodle and something doesn't seem right, then you need to let me know, right? So I do that so that you guys can always see what your grade is. So yeah, so that's basically, it's basically coded the right way in terms of calculating your grade. So like I said, if you have questions, let me know about that. There was somebody, or maybe a couple of people who asked about their grade, but so you always can see your grade. Any questions about that, guys? No? So like I said, I think I'm pretty much up to date with grading. I grade your exam 2. I will now grade your chapter 9 homework, and then I'll get 10 and 11 done. You will have one more lab. I think I told you guys this last week. I will put up one more lab. So let me share my screen just to show you these things as we go. So I'll put up one more lab. There'll be a short video demo, and there's some other things to go along with it, and an assignment. I'll put that up probably by Thursday. And that will be the last one. So it'll be due next week. And then you will have one more lab quiz / exam. That's all multiple choice again, which includes the mitosis and meiosis lab, and then this next lab that I was just referring to. So let me share my screen. Next week is the last week of class, right? I have, you guys can see my screen, right? So just a couple things, I guess, in terms of, right, so where we're at right now, last week was modules 10 and 11, right, which was chapters 9, 10, and 11. So now the homework and everything is due for that. So like I said, I'll review that. The exam 3, which is chapters 9 through 11, will be available starting on Friday. I may even have it up by Thursday, and I'll just always, like I always do, I'll shoot you guys an email just to say that it is available, okay? So I will, like I said, I was gonna do maybe a little bit of a review of chapter 9. I'll go over the homework questions and go over the quiz, I guess. That way on Thursday, I don't have to do 9, 10, and 11. I can just do a review of 10 and 11. And then you guys will be doing that next exam, okay? So that's that. And then the next module, right? Module 12 is this week, and then I have modules 13 and 14 as next week. But basically the 3 chapters that are left are chapter 12, which I'll put up a recording of that this week, and chapter 15 and 16. Okay, so the final exam will cover those chapters. I believe I also have some additional topics that will be on that final exam. So let me just, before I say the wrong thing, let me just double check that. Usually, and I mean, it's on the review sheet in terms of exactly what it is. So it's not cumulative. I guess I should probably, okay, so this is chapter 12, 15, 16. Right, and then and just be aware that there are some additional topics that will be on there that are listed here. So particularly plasma membrane and also biomolecules, okay? But again, otherwise, the final exam is not cumulative, all right? So like I said, next week is our last week of classes. I have the final exam as scheduled for Thursday, June 25th to Sunday, June 28th. Grades are due. So really technically this class ends that Friday. So I just gave you guys a few extra days to take the final exam if you need it. Grades are due by the 30th. So everything has to be in by June 28th, including that final exam, so that I can get everyone's grades together. The other thing is they sent this weird email to us that Moodle was going to be down on Friday, the 26th. Though we sort of questioned it because I don't know what that means, a whole day, and it's the last day of classes. So anyway, just to kind of be aware, if you try to get on that Friday, June 26th, and something's not right, it may be because of that, okay? So anyway, that's why I'll make sure that that final exam is available Thursday. And if there happens to be some issue on Friday, I mean, I'll know because I'll check Moodle. But so I just wanted you to kind of be aware. So if you can do it Thursday and just want to be done with it, then I would recommend that. Okay. In terms of that last lab exam, I have that lab exam as scheduled for Tuesday to Thursday. Okay, so I'll put that up as well. So all of the dates as they're listed on Moodle are correct. Okay, this lab assignment for that, so this DNA lab, there is a video that's posted. It's not my demo yet, so I'm going to put up a demo that sort of explains everything and kind of combines everything kind of in one. So I'll probably show the video in my demo, or parts of it at least, so then I can just kind of pause and explain things. Or I may just tell you, just watch that video. And then there is an assignment here. The actual lab report template's not in there yet. So I just wanted to make sure the assignment was up so that you guys knew there was going to be assignment. So once I do that lab demo, I'll obviously make sure there's an assignment in there. So basically it's going to be a Word document that you guys are just completing. Okay. So I have questions on there, and that's going to be that last assignment. Any questions, guys, about anything? Okay, so I wanted to make sure, like I said, that I updated you guys in terms of what's due, and make sure everyone knows that by the 28th, everything has to be finished. And then I will put your grades together. Also, that you can see your grade on Moodle. If there's a question, please let me know, okay? Sometimes there's little quirky things that go wrong with the grade book. I always obviously double check things a lot, and when I'm putting in final grades, I'll make sure everything is correct. But sometimes there's just a weird setting or some assignment that's not clicked on, and I just have to fix it. So please let me know. All right, so in terms of chapter 9, right? Chapter 9 was on cell cycle. Okay, so this assignment was due at 11 o'clock today. The two questions, pretty basic, right? Major events that occur at each stage during mitosis. You also are covering that sort of in your lab assignment, and in the lab exam, I'll show you pictures, and you should be able to identify cells and the various stages of mitosis. So I'll pull up the chapter 9 lecture just to show those pictures, because it's kind of weird to just talk about that without any anything to look at. But remember, the book does break down the stages of mitosis by having like a late, I think it goes like early prophase, late prophase, and then prometaphase. It sort of adds in these sort of intermediate stages. I usually like to refer to it as just prophase, metaphase, anaphase, and telophase, okay? And keeping with those standard phases, and I sort of explain that in the recorded lecture. But just so you know, it depends on what you kind of look at in terms of how it's broken up. And then the second question talks about proto-oncogene and tumor suppressor genes, and how these contribute to the development of cancer. So let me just quickly pull up chapter 9. I'll sort of answer those two questions. If anyone has any other questions pertaining to that, then let me know. And maybe I'll quickly go over the quiz as well, but let's do that first. So in terms of the stages of mitosis, it's straightforward. You probably did this in high school, right? I mean, it's pretty straightforward. What am I doing? Oh, I gotta go get the, all right, so what was it? Major events of mitosis and tumor suppressor and proto-oncogenes. So let me just grab that, the PowerPoint real quick. All right. It's a coming. Any questions, guys? Everyone doing all right? All right. I don't know if you guys saw too. Did she send the president sent out? The president of the college sent out some updates regarding kind of what some of the plans are for the fall semester. Did you guys see that? Did they send it to everyone?
Yeah, I saw it, but like I didn't like. What was she saying?
Yeah, I know. It's kind of like a lot of words that didn't really say much. I mean, think bottom line, and especially for us, for biology, is that, you know, it's going to be a combination of sort of online stuff and in person, particularly for us. And depending on how many students can be in one classroom at once, like for lab, we'll definitely be live. Like for the lectures, you know, I have them sort of recorded already. Probably that's the model that we'll use in terms of sitting and listening to a lecture. Those will probably be recorded, but when we come into class, we'll be doing more active types of learning kind of things, and things that we can maybe do in small groups That's the way that our department's gonna run it, but it kind of depends. There may be some classes that are all online. So yeah, it didn't really provide much clarity. The other thing though is the schedule, that the semester is going to be more compact, right? So the semester is going to end November 25th. So that Wednesday before Thanksgiving or whatever, when we normally would go to Thanksgiving break and then come back, the semester will end then. And then, so there's a longer winter break because they're trying to prevent, and I know a lot of schools are doing this actually, because I was in a meeting with a bunch of other schools for something. And a lot of them said the same thing, that they're getting rid of all the breaks so that then everyone just goes, on the break and then comes back for a second semester. So that was probably the biggest thing, but yeah.
So does that mean that we're starting earlier or no?
Not really. We were due to start the 24th anyway, August 24th, right? But she got rid of the Columbus Day break that we usually have and the Labor Day break. And normally we would only have really one more week, which would be finals week. So it's not really cutting the semester short at all. So it kind of, it makes sense. And I think, everyone's going to be happy to at least be back, live for as much as we can. I know, like I said, for us, because of labs and stuff like that, and because of hands-on things that you need to learn, we will be live. I know PA program, they're definitely going to be in, the simulation labs and stuff like that. Same thing for nursing. So for science and those types of programs, I think probably will be on campus maybe, or in class more than others maybe, but I don't know. My plan is to be in class as much as we can because it's way better. But there will be a little bit of mixing and it'll be a little bit different. But so I think the biggest thing was the schedule. In terms of what actually is going to happen, I think that sort of is going to be, each department will handle it a little bit different. But I told you kind of how we're going to handle it. So most of the in-class stuff will be doing stuff, not just listening to me talk or something like that you can do on your own. Anyway, I just want to make sure you guys, if you guys saw that. All right, so let me pull up that chapter 9 real quick. Just to kind of, like I said, address those two things. So one quick thing, and again, this wasn't one of the questions, but in terms of cell cycle, right? Remember the whole cell cycle is this. Mitosis is included in it. You have interphase, right? And then the mitotic phase. So understanding the cell cycle in terms of the phases of the cell cycle, remember, again, two main phases, interphase, and then the mitotic phase. Interphase is then broken up into these three phases, right? G1, S, and G2. And so any cell that is actively dividing, we say is sort of in the cell cycle, okay? And really the cell cycle is just sort of the life cycle of the cell, okay? If you notice here, you can have cells that are in G sub 0, and so you would think of those as cells like, neurons that don't, in general, there are neurons that do, there's a population of sort of stem cells that exist in the brain, but regardless, if we talk about cells that don't normally proliferate a lot or divide a lot, they're kind of in this quiescent phase here, okay? So basically cells, when they're in this interphase part, they're preparing to divide, right? They're gonna, in S phase, gonna replicate their DNA, and then go through another little period sort of growth where they're then gonna actually make sure all of their organelles are duplicated and things like that, and make sure they're ready to go through mitosis, okay? And also you should be aware of these checkpoints that are labeled here, G1 and G2, and this sort of spindle assembly checkpoint during mitosis, okay? So that's the overall cell cycle. So, I easily, on the homework question, could have asked you about, tell me the, the phases of the cell cycle, or the phases of interphase, and what's happening at each one of those phases. So you definitely should be really familiar with that, guys. All right, in terms of the cell cycle in general, right, I gave you a little bit of sort of kind of general, kind of broad explanation of controlling the cell cycle, okay? So actually, I guess I'll talk about the second question first. So I want you to understand why it's important that the cell cycle stays under control, right? If cells divide out of control, because that cell cycle is misregulated, it leads to things like cancer, right? Because cancer cells just divide and divide and divide, and they basically can ignore all of these sort of things that are there to slow it down, okay? And so this was just giving you an idea of some different types of signals, external signals, but there are also internal signals, and there's a lot more detail that we could talk about here, but I just wanted you guys to have a little bit of a general kind of just the idea that it is tightly controlled, okay, by different types of signals. And if it's not, it leads to cancer, right? And the formation of tumors. This process here, apoptosis, is the opposite of that, right? That's programmed cell death. So if a cell here, you know, if there's a problem detected that can't be repaired, the cell would go through apoptosis. Along the lines of the cancer cells that I was talking about before, if even though there's a problem, the cell doesn't undergo apoptosis, again, that leads to cancer. And so a lot of times with cancer cells, they have problems with, you know, these checkpoints, or again, being able to slow down the cell cycle, but also the decision as to whether to go through cell death or not. Cells that should go through cell death won't, right? And they'll have some sort of inherent issue with the DNA. And even though they do, they're going to keep dividing, right? So again, that leads to more problems and more dysfunction, okay? So you don't want dysfunctional cells to divide, right? Again, those are, think of cancer cells as these dysfunctional cells that keep dividing when they should have went through apoptosis. So in terms of, you know, genes that control the, right, that are involved in regulating the cell cycle, that's what these tumor suppressor and proto-oncogenes are, okay? So they're kind of the opposite of one another. So I'm gonna go back to the phases of mitosis, but let's just talk about that real quick, okay? So a proto-oncogene is a normal gene that everyone has, right? That codes for a protein that normally promote the cell cycle, right? Or promote cells to divide. And there are times when you want cells to divide, right? If we're talking about growth and repair mechanisms, you want to be able to promote cell division, okay? If one of these proto-oncogenes becomes mutated, right, there's some sort of damage, it leads to them becoming an oncogene, okay? So an oncogene is a dysfunctional proto-oncogene, meaning that mutation causes this proto-oncogene to be on all the time. So then the cell cycle is always being stimulated, okay? And so not good, right? You want to have the ability to turn up the cell cycle or increase cell division, but you also need to be able to turn it down, right? On the flip side, a tumor suppressor gene is a gene that will code for a protein that normally inhibits or slows down the cell cycle, okay? Now, if there's a mutation in a tumor suppressor gene that basically causes it not to work, then the cell cycle would be sped up. Okay, so you can have mutations in either or, but the mutation causes different things, right? So the mutation in a proto-oncogene would cause it to be on all the time, and that could lead to cancer, right? In a tumor suppressor gene, the mutation would cause that protein to not work at all, right? To be dysfunctional. Well, I mean, dysfunctional could be, so it would cause it to not work, put it that way. And then you would lose that inhibition, right? And then that would lead to increase in cell division, okay? So that's how those contribute to cancer. And again, these tumor suppressor genes and proto-oncogenes code for, you know, things that are involved in regulating the cell cycle. So I kind of, in the beginning, I talked about, you know, like signals and things like that, and growth factors, and maybe receptors for those growth factors. Also, there's certain proteins, these internal signals, these cyclins, and lots of other proteins that are involved or associated with them. Tumor suppressor genes and proto-oncogenes code for those types of things, okay? So if there's a mutation in anything related to the regulation of the cell cycle, it could potentially lead to cancer. And there have been, you know, there's certain cancers that are implicated, or certain genes that have been implicated in certain cancers. And those are, you know, those are mutations in tumor suppressor genes or in proto-oncogenes. All right, so that's the relationship there that I sort of want you guys to understand. The first question just asked about the phases of mitosis, okay? in terms of, again, what's happening. But remember, I like to just say prophase rather than dividing it up into early and late prophase. But one of the things that you'll start to notice at the beginning of mitosis is that, in the very early prophase or even before prophase, the DNA sort of looks like right, if you look at a cell under the microscope, sort of looks like a bowl of spaghetti. Those chromosomes aren't distinct or separate, right? But as prophase starts to continue and as you get, move into mitosis, you can actually see the chromosomes as looking like more individual chromosomes, right? You can start to see that the spindle starts to form, okay? And also remember that you should be able to identify these stages from pictures, right, on that lab quiz, both from animal cell and also from plant cells, okay? So this is a whitefish blastula, right, an early embryo of a whitefish, and then this is an onion root tip cell, okay? All right, so anyway, and then if you move on, right, so again, we sort of add this little extra phase sort of in between prophase and metaphase. What you notice now is the spindle is more elaborately formed, right? The spindle poles have moved to opposite ends. You can see here it mentions the kinetochores here, and that there's kinetochore spindle fibers that start to attach basically to the chromosomes, right? And then as you move on, right, metaphase is easily distinguishable because all of the chromosomes are lined up in the center, right? One other thing to point out here, if you look at these chromosomes, they have that characteristic X sort of shape. Why? Because they are duplicated. Remember, you only see these sister chromatids in this sort of X shape of the chromosomes when the chromosomes are duplicated. Remember, they are duplicated in S phase because the cell is getting ready to divide, right? You start with a parent cell and you're going to end up with 2 identical daughter cells. If the cell did not replicate its DNA before mitosis, those 2 daughter cells would end up with half the amount of DNA. That's not going to work. You need all the chromosomes, right? You need all the DNA to be a functioning cell. So anyway, here in metaphase, you have those chromosomes lining up. And I'm obviously leaving out some details here. You guys can read through them. And then in anaphase, right? Again, if you look under the microscope, you can clearly identify this cell as being an anaphase. Now the sister chromatids separate from one another, right? They're moving towards opposite poles. This is going to be one cell, and this is going to be one cell, right? Finally, in telophase, you start to have the nuclear membrane reappear, right? And basically, again, here's one cell, here's another cell, and then this is showing you this cleavage furrow that forms. Again, now, remember that mitosis refers to nuclear division and what's happening with the chromosomes in the nucleus. And remember, cytokinesis is the formation of this cleavage furrow and basically the dividing of the cytoplasm, okay? So remember that cytokinesis is not part of mitosis. It happens along with it, right? It has to happen to divide those cells physically, but mitosis refers just to what's happening inside the nucleus and what's happening with the DNA, okay? And so that's basically it. So the first question was about the stages of mitosis. The second question was about the tumor suppressor and the proto-oncogenes. Okay, so I mean, I hit some high points. Again, I left some things out from, from chapter 9. If on, Thursday when we meet again, if you have some other questions, ask me, but I'm not going to review chapter 9 again. What I am going to do before we finish this meeting, and I'm sorry, I don't know why I'm so dark. Oh, that's because I don't have my lights on. What I am going to do is let me just go over that chapter 9 quiz quickly, okay? And then that's it. All right. My kids are yelling in the background. Awesome. All right. So let's go over that chapter 9 quiz. Okay. So only 10 questions, super quick. So like I said, so then that way on Thursday, I don't have to go through all 3 chapters. I can just review chapters 10 and 11. The 2 main phases of the cell cycle are, I already reviewed this, right? Interphase and the mitotic phase. Okay. DNA is replicated during which stage of interphase? It's S stage or S phase. I don't know why I have that written as stage. It should be phase, but whatever. Same difference. Right? S for synthesis. During the blank stage of interphase, the cell grows and makes its final preparations for division. And so that's G2, right? Remember, you have G1, S, and G2. G2 is right before the mitotic phase. Oy, oy, oy. Sorry, got lots of noise here. All right, sorry. Somebody's cutting the lawn outside. The cell divides during which stage of the cell cycle? It's during the mitotic phase, right? Mitosis is cell division or nuclear division, really. If DNA damage is detected at this cell cycle checkpoint, the cell is sent into apoptosis. If there is no damage detected, the cell will then enter into S phase. And so the answer to that is that G1 checkpoint, which is probably the most important checkpoint. Basically, a cell's not going to proceed through that checkpoint if things aren't right? It doesn't make sense for the cell to waste its resources and replicate the DNA if it's not then going to divide later on. All right. Okay, blank are proteins associated with DNA that aid in packaging DNA into chromosomes. Okay, so these are histones. So also in that chapter, I went over sort of the packaging of DNA, right? And we talked about chromatin, right? Euchromatin and heterochromatin. Talked about a little bit about nucleosomes, and essentially that's just the DNA wound around these histone proteins, okay? How many chromosomes do humans have? 46, right? Remember, we have 2 sets of 23, 46 chromosomes total. In blank of mitosis, the chromosomes line up in the center of the cell. We just went over that, right? And that's in metaphase. In blank of mitosis, the 2 sister chromatids of each chromosome separate and move towards opposite ends of the cell. That is anaphase. And again, when you're sort of looking at those cells to identify them during that lab quiz, they have this sort of V shape, right? They're pulling away from one another. All right, last question. Blank are proteins that are involved in regulating the cell cycle. Okay, that answer is cyclins. Okay, so cyclins are a family of proteins that are produced by the cell that are involved in regulating them, right? And so if there was a mutation in a gene that codes for one of those proteins, it could potentially lead to the development of cancer, right? Because it could lead to misregulation of the cell cycle. All right, and so that is that for that quiz, and that's pretty much it for reviewing chapter 9. All right, so let me stop my share. Let me move this out of the way. All right, guys, so that's pretty much that's it. I mean, I reviewed chapter 9. Like I said, be on the lookout for one more lab, right? I'll call it the DNA lab. We're doing a DNA extraction. And then also I'll have a demo on DNA electrophoresis, and I'll also demo how to use a micropipette. So it's kind of those 3 things. And then there's a little bit of an application of DNA fingerprinting that's in there because you could potentially, I mean, it's sort of probably done in a more automated way now, but you could potentially can use, you know, basically running a gel and looking at the different the DNA banding patterns between, you know, evidence at a crime scene and individuals to, you basically can use it in forensics. Okay, so I just like to always give you guys a little bit of an application. But that's pretty much, that's it, guys. Does anyone have any other questions? All right, guys. So that's it. I'll see you guys on Thursday. Just make sure you keep keeping up with the work.
Thank you. Thank you. Bye.
Thanks, Lisa. Bye, guys. Thanks.